next on our list is Bill Martin. Bill is a professor at the Heinrich Heine University in Düsseldorf, the head of the Institute of Molecular Evolution. I'm looking at the list of speakers that we've had on this first day. It really is a star-studded attraction. So thanks, Bill. Over to you.
Well, thank you very much for the organizers for this kind invitation. I'll try to keep you entertained for about a half an hour to 45 minutes about my thoughts on the origin of life from the perspective of a biologist and the perspective of comparative physiology. So as I see it, life is a chemical reaction. Just a second, let me try to get that. Yes. Life is a chemical reaction. If you don't believe me, take a plastic bag, put it over your head and hold it real tight for about a minute, and the physiological reaction of your body will attest beyond all reasonable doubt to the view that life is a chemical reaction. We have to have energy, and we get that energy for the life process from the respiration of fats, amino acids, and sugars in our mitochondria using oxygen as a terminal acceptor. That generates an ion gradient that generates ATP, and it's that ATP that runs all other life processes. So from the standpoint of physiology, if life is a chemical reaction, that's not a definition, it's an observation. Then what I want to know is from which chemical reaction did it start? All right, so that's where I come into this issue. From the physiological perspective, life is a chemical reaction. There has to be some naturally occurring chemical reaction from which it started, because life is an energy releasing process, and that means that the initial process from which it arose also had to be energy releasing. So in evolution starts right here. This is Darwin's famous tree figure from his book, where we have natural variation and natural selection, the only two principles that he needed to explain the observed diversity in the then known living world. At each generation, we've got variation. Okay, those are these different little lineages coming off here from every generation. Those are he didn't have a mechanism, of course, for this natural variation. He just observed that it occurred because he went to dog breeding shows and pigeon breeding shows. And then, of course, his big leap, some of these variants were more fit to make it into the next generation. And so we can explain the evolutionary process. And what we're doing today is trying to extrapolate from up here, the things we can see today, back down to here, the things that existed at the origin of life. And that works very well for living organisms, but there's a problem with Darwin's principle as it relates to very early evolution, and that is that Darwin's mechanism works much better if you already have something that is alive. If you're down here, before we have things that are alive, we just have chemicals and rocks and water on a rocky planet. This transition becomes very difficult. That's what we'll be dealing with today, hopefully in a way that will make it more imaginable for those of us who are watching. So this is another view of early evolution. This is Ernst Haeckel's famous tree from 1866, only 7 years after Darwin published Origins. And this tree also has evolutionary lineages, this time not schematic, but actually labeled. And Haeckel could also draw each one of these organisms masterfully. And he put the whole tree together in a system that's not too terribly different from what we see today. But also, Haeckel was cheating a little bit, because he started down here with something that's big and strong and well differentiated. There are no roots on this tree that would help connect it to inorganic substances. But he did mention in one of his lectures that maybe life started from CO2 and ammonia and urea and inorganic salts. This guy here, Konstantin Merezhkovsky, was actually very explicit about the or evolutionary process, or the chemical process that gave rise to life. In his tree, published in 1910, you can see that there's something going on down here, some chemical process going on, giving rise to this most simple biococci, then micrococci. These give rise to eukaryotes via symbiosis into in his view, a blob, kind of a eukaryotic precursor cell, and then evolutionary processes on down the line, including endosymbiosis. But the main thing is that Merezhkovsky was concerned here about chemical processes. I don't have this translated here, but I do have it translated in Biosystems that appeared this year, Merezhkovsky's 1910 paper. What he was saying about the nature of the first cells, just reasoning by comparative physiology, is that they had a minimal size. Okay, they were too small to be seen under a microscope, the very first cells. They had no organization. What he meant by that was that they were not eukaryotes that had obvious cellular organization. They were prokaryotes. They had the ability to survive high temperatures close to the boiling point. Well, that's thermophilic origins in 1910. They had the ability to live without oxygen. Well, they were anaerobes. That's a completely modern idea, and we still believe that today. They had the ability to make proteins and carbohydrates, the latter without the help of chlorophyll from inorganic substances. That, ladies and gentlemen, is chemolithoautotrophy in the modern sense. And this is 1910 by pure reasoning. It's a very interesting paper. I can recommend the translation. And they were also highly resistant to acids and bases and sulfur compounds and various poisons, toxins. So that's chemolithoautotrophic origins in 1910. The idea has been there a long time from the standpoint of physiology. So where might that have come about? We'll just make a big leap here to Mike Russell and Deb Kelly, because they're the people who got me interested in hydrothermal vents. Mike, because of papers that he wrote and papers that he sent to me in 1998, and he wrote, among other things, we proposed that life emerged from growing aggregates of iron sulfide bubbles containing alkaline and highly reduced hydrothermal solution. These bubbles were inflated hydrostatically at sulfidic submarine hot springs sited some distance from an oceanic spreading center 4 billion years ago. Now, when I read that, I said, what is he talking about? I had never heard of any of this stuff. I'm a physiologist working with enzymes and substrates. Deb Kelly then found exactly the kind of vent that he was talking about, called Lost City, the first kind of its first of its kind. When they were on a cruise in the Atlantic, and it contains alkaline, highly reduced with roughly 10 millimolar hydrogen, sulfide-bearing water, and they form bubble-like microcompartments, just like Mike was saying. Well, what's the rock that Mike got so excited about? Here's a picture of that rock, published by Mike and Alan Hall in the Journal of the Geological Society of London in 1997. And what you can see is a chimney here about the size of a finger or a laser pointer if we were holding a live talk. And here, botryoids, little grape-like, bubble-like compartments or structures that are being formed as hydrostatic pressure is pushing water up out of the bowels of the earth. We're looking at the surface of a fossilized hydrothermal vent, 360 million years old, that Mike found in silver mines in Ireland. And when he cut it open, they're hollow. They have internal structures. These microcompartments have internal structures. Now, that's very interesting from an origin of life standpoint, because if you look at it from the biochemical or physiological standpoint, the first thing that happens and has to happen in any of this chemistry is that the products that are synthesized have to be concentrated around their site of synthesis. Otherwise, they are going to diffuse off into the environment and will be worthless for the process of evolution if they diffuse off into the ocean. So there has to be some naturally existing compartmentalization function provided by the site where life might have arisen. These inorganically formed microcompartments do exactly that. They have inorganically formed walls made out of iron sulfur minerals that are generated by the hydrostatic pressure in the hydrothermal vent. In this case, it's It was iron 2 rich water meeting sulfide rich water at the bottom of the ocean. And the other function, so these compartments fulfill two functions. One is compartmentation. The other function that they fulfill is catalysis. Now, to the chemists in the audience, I don't have to say this, but for some of the biologists or other disciplines, I might. But let's pretend that, okay, why are these transition metals important? Let's pretend that we've got a transition metal here about the size of a basketball. That could be iron or nickel or molybdenum. And what these transition metals have, they have what are called d orbitals, okay? They have d electrons, and these d electrons have very interesting geometries that can undergo interesting hybridizations. And they can make metastable bonds with carbon and nitrogen so that the carbon and nitrogen can react, and then the carbon and nitrogen does some reaction up here, and then the d orbital is pulled back in, the carbon or the nitrogen leaves the catalyst changed, reacted, but the catalyst itself is unchanged. It's a catalyst. The important thing is that the property of catalysis can be modified by chemists in the way that they coordinate these transition metals with special organic cages or any number of things, or inorganic structures. But the property of catalysis is a property of the elements themselves. There's nothing that we have to do to make those elements catalytic. It's a natural property. So what we've got are naturally catalytically walled microcompartments being formed at a submarine hydrothermal vent 4 billion years ago. And they're able, in theory, to do some chemistry. And we'll see in the course of the talk what kind of chemistry I'm thinking about. So, okay, we're not talking about black smokers. All right, the black smokers are, of course, the famous ones that were discovered in 1978. They're very hot, 400 degrees. That's too hot for life. And they don't have the right chemical composition, we think, to bring forth the chemistry that would be typical for life. We're thinking more in terms of Lost City. This is a picture provided by Deb Kelly. Thank you, Deb. The spires at Lost City get up to 60 meters high. They're at least 30,000 years old, probably closer to 100,000 years old. And the water coming out of these vents is alkaline. It's warm. It's about 70 degrees. It's very hydrogen rich. And hydrogen, as we've heard in Shino's talk and also in Barbara's talk, is a source of microbial energy. Okay, it's energy in electrons. You have to have an acceptor, and what that acceptor is, we'll get to in a minute. So that's also an interesting property of the alkaline hydrothermal vents like Lost City from serpentinizing systems. They're stable over long geological timescales, whereas the black smokers have lifetimes on the order of tens to hundreds of years. Okay, so also the Lost City compartments, if you cut open a section of Lost City, it also forms large systems of naturally forming microcompartments that can get very large. Think about that, over 60 meters. It's a system like a chromatography column, huge surface areas where lots of stuff can interact. Okay, the reason that the surfaces are important is because of catalysis. So that gave rise to a paper by Mike and myself in 2003 that appeared in the Philosophical Transactions of the Royal Society of London, the world's oldest continuously published journal. And I drew this figure, and this figure goes like this. All right, so on the early Earth, we've got hydrogen coming out of the bowels of the Earth via serpentinization, and this is interfacing with CO2 in the ocean, all right, because there's lots of CO2 in the ocean. And why that is because of the Earth, the moon forming impact. And if my movie will work properly, we'll be able to see that later. That's a redox couple, and there are, being a biologist, I know that there are organisms that live from that redox couple. That is, they extract their energy from making their ATP by transferring electrons from hydrogen to CO2. So that's a redox, that's chemical energy. Right? Microbes can use that. And that means that there should be enough energy for synthesis of reduced carbon compounds, call them thioesters or amino acids or sugar or bases, just some sort of organic chemistry being catalyzed by these transition metals at the vent. And because they're compartmentalized, they don't diffuse off into the ocean, and therefore they can undergo transitions to higher complexity in the model, much higher complexity, and this can go on for hundreds of thousands or 100,000 years perhaps, and maybe we can get to something that's a very complex system, similar to what maybe Steve Benner was talking about earlier today, but made the way that biology makes things and not the way that chemists make things. So that would give the model contains a last universal common ancestor that is not free living. It is contained in the compartments within which it was formed. Integral to the model is the idea that the iron sulfur centers in modern proteins are not inventions of the biological world. They are just inventions of the protein world, that when life got to the stage that we had genes and proteins, they could incorporate these iron sulfur clusters into the cysteine centers of these of polypeptides, so that the iron sulfur clusters became, one, genetically specifiable, and number two, portable, so that life could escape from the vent. All right, so this is very similar to the RNA world. RNA cofactors are ancient, iron sulfur clusters are ancient. And the further in the further process of this evolutionary scenario, hydrogen and CO2 continue to interact in a stable disequilibrium, so that we get continued synthesis of more complex organic compounds. Then at the very end, we get the compartmentalization. That is, the cells learn to compartmentalize themselves from the environment by themselves, with the help of information that is contained within genes. We've already got the origin of the genetic code, which we just glossed over, but we know that it occurred in the last universal common ancestor because all cells use it. How that occurred is still debated, but everybody knows that it did occur in that last common ancestor. That's also part of this model here. The fatty acid esters of the bacteria are unrelated, both in their biosynthesis and their chemical synthesis, to the isoprene ethers of the archaea in the lipids. That's independent evolution. The same thing for the S layer of the archaea and the peptidoglycan layer of the bacteria, unrelated pathways, independent evolution. And that allows them to escape, provided that they are able to satisfy their energy needs from H2 and CO2 alone. And that's part of what the rest of the talk will be about. So where does this hydrogen come from? For those who are not familiar with it, it comes from a well-known geochemical process called serpentinization. It doesn't have a very sexy name, but it's a very important process. And Wolfgang Bach and colleagues wrote it this way. It's iron magnesium silicates in the Earth's crust, in ultramafic rocks. I guess ultramafic means low silicate. This is geospeak, but iron, the main thing is it's iron 2. Small amounts of iron 2, substoichiometric amounts of iron 2 in these iron magnesium silicates. And this serves as an electron donor to reduce water. The iron 2 is oxidized to iron 3. This is magnetite. It has 2 atoms of iron 3 and 1 of iron 2, along with the oxygen here. And hydrogen gas is produced as an end product. Okay, this comes out, the hydrogen gas comes out from the vent. So basically what is happening is that the Earth's crust during serpentinization is reducing water to hydrogen gas. The reason that this process takes place is because it releases energy. It's exergonic. There is no extra energy that has to be added, no UV light, no extra gravity, no antimatter, no anything. It's just straight chemical reaction. And you ask, oh, wait, where did the energy come from that's driving this reaction? And it comes from the melting of crust through subduction, then it mixes with the mantle, and then it becomes more reduced, and then it comes back up, and then the more reduced form, it can become more oxidized. And this process, these are called Wilson cycles, and they go on and on and on. And this process has been going on as long as there have been water on Earth, because there's no way to stop it. The product serpentinite is this. You see it in banks and nice hotels. It's the green rock, and the veins are where water was going through. And the reason that this is interesting to me is because this process generates hydrogen, and it's starting to look very similar to something that looks familiar from microbial physiology. All right, so acetogens are bacteria. We heard a lot about acetogens in Shino's talk, and we heard a lot about acetate in Barbara's talk. They live from this reaction here. Hydrogen plus CO2 gives us acetate and water, and that reaction is exergonic. It releases about minus 104 kilojoules per mole. That's definitely enough to pump protons, and that's how they live. Methanogenesis is a little bit more exergonic, minus 100. The 1 kilojoules per mole under standard physiological conditions because the product is more reduced. And to the level of the energy-rich thioester, CO2 plus H2 plus the thiol-CoA gives you the thioester, acetyl-CoA, and water, and that reaction is also exergonic. This is something that Georg Fuchs has been saying for 35 years, and we haven't been listening closely enough. A few have, but it's important. The organisms that use this pathway release energy, and in the process of fixing CO2 that they can conserve in ATP synthesis. All right, so how does this work schematically? We're not going to go through biochemical pathways here, but just in general, the pathways are related in terms of their chemistry. You got CO2 to formal to, and actually this is old. The methanogens also make formate. It just doesn't leave the protein. CO2 to formal, formate to formal, to methanol, to methylene, to methyl. Okay, these are different reduction steps. They occur on a cofactor. This is tetrahydrofolate. This is tetrahydromethanopterin. They're both pterins. They're just very standard sorts of cofactors. And this methyl group is then donated to a very conserved protein that is common to both pathways. That's CODH, ACS, carbon monoxide dehydrogenase, acetyl-CoA synthase. Now, the two methyl synthesis pathways are not related. Okay, the enzymes are not related. So sometimes I get this question, Dr. Martin, how can you possibly suggest that these two pathways are ancient, which is what I am suggesting, if the enzymes are not related? They're independently evolved. Well, what we're saying here is that the chemical reactions are older than the enzymes that catalyze them. Okay, and there's nothing spectacular about that. That's standard biochemical reasoning. Enzymes do not perform feats of magic. They just allow reactions to go faster. They just accelerate reactions that tend to go forward anyway. So two things to say. Number one, these same sorts of reactions take place at Lost City. Yoria Proskorowski et al. And colleagues from Deb Kelly's group showed that in a very nice paper in Science in 2008. And this reaction here, this acetyl-CoA synthase carbon monoxide dehydrogenase reaction, that's very interesting because it gives rise directly to the energy-rich thioester. So this is very central in the energy conservation principles of these organisms. What's going on there? And what's interesting is that, okay, this is iron sulfur chemistry, and Günther Wächtershäuser and a lot of other people tried for years to get iron sulfur minerals to reduce CO2. Some people are still trying it. But if you look at biology, that's not going to work really, because CO2 reduction is a 2-electron reaction. CO2 takes a hydride. Okay, that's a proton with an electron pair. That's how CO2 reduction works in biology, without exception. There are no exceptions. There are some radicals in biology, but not in CO2 reduction. All right, it's always 2-electron transfer. And iron sulfur is undergoes 1-electron transitions from iron 2 to iron 3. Well, that's not very good. And I was looking at the reaction mechanism in a paper by Steve Ragsdale, and it caught my eye. I said, my goodness gracious, why is this not working? How can we get past go and collect 200 at the CO2 reduction step, the entry into all of organic chemistry? Because that's what there was. There was CO2. That's the ancestral state of carbon on the planet after the moon forming impact that melted the planet. Right? It's magma. Right? All the carbon that was secreted there, it's CO2. All right, so that's what we've got to work with. Well, in the enzyme reaction mechanism, all right, they've got this reduced nickel here at the active site. CO2 binds to that and bends. And when CO2 bends, it's activated. That means it's getting ready to make new bonds. And that's exactly what happens here. It actually takes 2 electrons off of this iron sulfur cluster and becomes carbon monoxide. Water is also produced in the process. The other oxygen here on CO2 comes off as water. That gives us an oxidized cluster nickel 2, and then 2 electrons from ferredoxin regenerate nickel 0. Nickel 0, it's a native metal. Oh, interesting. 2 electron reaction, nickel 0 to nickel 2. Will that work in the laboratory? So the idea started in 2002, and in the meantime, it's 2009, and then by that time, it was 2015. And we started to look, and I got this outstanding PhD student, Dr. Martina Preiner, who's now at the University of Utrecht. My team, led by Verena Zamorsky and myself in Düsseldorf, and we got some help from Harun Tusuz at the Max Planck for Coal Research. In Mulheim, you might say, Max Planck for Coal Research, what is that? Well, that's the place where the Nobel Prize went about a month ago for chemistry, because they work on catalysts. Harun works on inorganic catalysts. Professor List works on organocatalysis, that is, amino acids that are catalytically active. Chris Greenwell is at the University of Durham. We tried to get some reactions to work there, and then Joseph Moran at the University of Strasbourg. We started because I was talking to Tom McCollum with a mineral called awaruite. I said, Tom, are there native metals in these hydrothermal systems? He says, Bill, awaruite is in all of them. It's iron, it's nickel 3 iron. It's a natural native metal. Ubiquitous in serpentinizing systems. It's generated by H2 reducing iron 2 and nickel 2 species. Okay, it's deposited there, usually in microcrystalline form. These chunks that we see here, these are meteorites. That's just to make it visible, because the nanoparticular awaruite is difficult to see. So with Harun, we were able to, with CO2 and H2, thinking that the nickel would do the job. We got some CO, just a little bit. With iron nickel carbonates and Chris Greenwell and Durham, 400 degrees, we got a little bit of methane, but that wasn't anything to write home about. Then Joseph Moran started showing that native iron particles would catalyze 6 out of 11 reactions of the RTCA cycle, now using CO2 as a starting material. That's interesting. And then another paper, Varma et al. In Nature, Ecology, and Evolution 2018, they found that CO2 and iron 0 would give us formate, acetate, and pyruvate. Whoa. That's interesting, because that's the backbone of carbon and energy metabolism of in organisms that use the acetyl-CoA pathway. So Martina and I started with nickel iron again, awaruite, 100 degrees in water using CO2 and H2, just like the microbes do. And overnight, without any other excess form of energy, we've got formate in 100 millimolar, sometimes up to 500 millimolar, half a molar formate we get overnight, acetate, 100 micromolar, ethanol, 50 micromolar, pyruvate, 20 micromolar, and methane. That's significant in our view that it was published, because what we're doing in this little reactor, we're adding hydrogen, CO2, water, and a catalyst, a native metal catalyst, 16 hours, 100 degrees. The backbone of microbial carbon and energy metabolism in acetogens and methanogens unfolds in front of our eyes. Okay, it's very natural. Furthermore, those are the only products we see. We don't see these among a slew of other stuff and tar. It is formate, acetate, pyruvate, methane, and methanol. Those are the products. It's a very specific synthesis. It's amazing. And I can't say enough about Martina Preiner and how she set that up in the laboratory. So this is just to show that the catalysts, when we analyze them by X-ray diffraction, they are unchanged after the reaction. They are catalysts. They're not undergoing oxidation themselves. They are activating the hydrogen. And this is the reaction pathway, the biological reaction pathway, redrawn from Georg Fuchs. CO2 and H2 formate, formal, This means here it's bound to a cofactor. This is a pterin cofactor here. This is a pterin cofactor here. It can also be an iron atom. This is an acetyl group. This is CO. This is the acetyl synthesis part of the acetyl-CoA pathway. But all of these intermediates are all cofactor bound in the pathway. They don't appear, you don't get free formate, you don't get free methylene or formaldehyde groups. These are the products that we get with our awaruite or magnetite or greigite catalysts. We get formate, we get methanol, which can be incorporated into the pathway. It's not a product, but it can be incorporated. Methane, acetate, and pyruvate. Those are exactly the products we get. All right. So that's very interesting. And that indicates to us that this chemistry is taking place on the surface of the metal. So that's what's driving all this, we think, chemistry, getting pyruvate. If you know your biochemical pathways, you will know that there is no compound in carbon and energy metabolism that is more central to all of biosynthesis in cells than pyruvate. All right, so that's part of this, right? And then we said, all right, so the basic reaction to get us going here works. Can we learn something more about LUCA down here at the in the last common ancestor? Okay, so we did some genetic stuff on genomes, looking for genes that are present in both archaea and bacteria, but not universally present. We knew those. Those are ribosomal proteins in the ATP synthase. We were looking for proteins that could be traced to the LUCA, but are not universal. And there are two ways to get that, either lateral gene transfer or vertical descent from LUCA and differential loss. And we differentiated those by looking for proteins that have this pattern, that is, the bacteria and the archaea are monophyletic, but the but the protein traces to LUCA, present in at least two groups and monophyletic. If we do that, we find 355 proteins. This is 11,000, that's 355. And what we found there were ribosome biogenesis, we knew that, RNA modification, translation, we knew that, DNA binding, yes, nucleic acid synthesis, energy metabolism, carbon metabolism. Whoa, wait a minute. Nitrogen metabolism, cofactor synthesis. We're getting a look at the physiology and habitat of LUCA. And as if you put this together in a scheme, this is what you get. You get a cell that's half alive. 355 genes is not enough to provide a full free living cell. It's able to harness ion gradients, but not able to generate them himself, but that's okay because the ocean is a little bit more acidic than the extremely alkaline water produced by hydrothermal fluid, pH 9 to 10. There's no methyl synthesis branch in the acetyl-CoA pathway, but there is the CODH reaction, the thioester reaction, and substrate level phosphorylation from the thioester. So this is, we think, how LUCA was living: ferredoxin, flavoproteins, corins, molybdenum, selenium, metals, and simple physiology. All right, so that's interesting. Can we learn a little bit more? So we have to have something new. And if we look at the overall setup of metabolism, it's not that complicated. We can do this. H2 and CO2 give us C1. These then acetyl units and pyruvate. Pyruvate goes on to alpha keto acids and the reverse citric acid cycle to make amino acids, and sugars go on to make cell wall and cofactors. Amino acid cells are 50% protein, 10% cell wall, 10% lipids, 20% RNA, and 3% DNA, and the rest is just metabolites. How many genes are actually needed to do this? Because this is the synthesis of all the cofactors that we know. Here are the pterins up here. Here's the corins over here, iron sulfur clusters. Here are the standard B vitamins over here. How many proteins? It's 400. This is published by Jessica Bimont and the team in Dusseldorf this year in Microorganisms. And it's a very interesting paper showing there's 402 reactions. But this figure here, we've made the map in such a way that we've got the starting compounds here. This is not published yet. It should appear soon if we're lucky. Hydrogen, CO2, ammonium, phosphate, water, and hydrogen sulfide. Those are our starting compounds. Everything else out here in this network is products. It's complete and it's 402 reactions. And what you see here is the answer to the question, is thermodynamics with us or against us? And so what you see, the green ones are minus 32 kilojoules or more exergonic. If Steve Zender is here, thank you, Steve, for pointing out the significance of thermodynamics. Very few reactions are actually strongly endergonic under standard physiological conditions. And so there's a natural tendency, these are exergonic reactions. The synthesis of amino acids, bases, and cofactors is exergonic. That's interesting. Well, how bad is it? And what conditions do we look at? Well, you can change the temperature, 80 degrees, 25 degrees. You can change the pH between 7 and 9. You can look at equilibrium or non-equilibrium conditions. In non-equilibrium conditions, what we do is we polarize all the reactions in the direction of biosynthesis and put more products on put more higher concentrations on the reactant side so that there are fewer products. And because this simulates the situation at the origin of metabolism where there were no products to push back. It was a flow forward synthesis. And if we do that, we find that actually 97% of the reactions in metabolism, up to 98%, are exergonic under the conditions of hydrothermal vents. Well, what about sources of energy? Well, we know that there are 77 ATP-dependent reactions there. We've got a source for substrate-level phosphorylation in this network. It's the synthesis of acyl phosphates from thioesters and phosphate. But what about the other ATP-independent reactions? Well, here we go. We've got pyruvate formation from H2 and CO2, and there's one reaction of that type. That's the acetyl-CoA synthase reaction, minus 57 kilojoules per mole. Ring formations, there are 35 of those, minus 10 to minus 25 kilojoules per mole. Decarboxylations, minus 20 kilojoules per mole, 30 of those. SAM-dependent alkyl transfers, folate-dependent acyl transfers, reductions, that is NADH-dependent reductions, or ferredoxin-dependent, or NADPH, or FAD-dependent, minus 28 kilojoules per mole on average in our network. Folate-dependent alkyl transfers, thioesters, acyl phosphates, and aromatics. This is often forgotten. The synthesis of aromatics is extremely exergonic because they're much more stable. Those are sources of reactions, sources of energy within the core. The energy at the origin of metabolism came from within metabolism itself. This is a really interesting insight. And it comes from just looking at the reactions of metabolism and polarizing them in biosynthetic direction and doing the delta G. And there are a lot of people who do not want that paper to appear. So let's do the movie, and that'll more or less be it. We want to get to the end here. Let's see. Can you still hear me? Can anybody still hear me? I have no idea whether anybody can still
hear me. We can indeed.
We're still here. Don't worry.
Okay.
Still here.
Okay. There are still humans out there. All right. So let's try the movie. It's not done yet. All right. This is the premiere. You're going to see where we got some problems, but it's a 5-minute movie on the origin of life, and then we're basically done. And it's the origin of microbes, because I'm not worried about definitions of life. I'm talking about the origin of microbes, and there is no debate on that definition. Recognize this. This is Theia. This is the moon forming impact. Okay. It's a big problem for the early Earth. It melts the Earth. We got problems with the animation because they didn't get the origin of the moon right. We're going to have to go back and work on that tomorrow, but it'll be another 2,000 euros, but they'll get it right. Because watch this, the moon just comes from nowhere here, right? No. We've got to do that over. So at any rate, it's a boiling ball of magma. That's important because it gives us differentiation. All the molten metals go to the core, and that gives us a magnetic field, and that saves the Earth's atmosphere, because the Earth's atmosphere starts to form here when that big ball starts to cool. And it's a CO2 nitrogen atmosphere full of water, gas. All of that stuff was transferred to the atmosphere because of the molten nature after the moon forming impact. All right, so it rained for 40 days and 40 nights. We got very deep oceans, twice as deep as today's, because there's about an ocean volume worth of water in the crust and mantle today, and that wasn't there at the beginning. And there's a little bit of meteorite impact for those of us who like meteorite impacts. Now, please, geologists, look the other way. It's a schematic. It's for school children and public lectures. It's hydrothermal vents at a spreading zone, and they've got a lot of magma going around over there. Those are the black smokers. We don't want those. This is what we want over here: a lost city type vent, an off-ridge vent at a serpentinizing site. And if we can part the waters so that we can look into the inside of this, there we go, we can see what's going on. Over here at the hydrothermal, at the spreading centers, at the black smokers, the water that's circulating through the crust is coming into contact with magma. That's too hot. That's why they're not good. And they also have a short lifespan. You see that one disappearing over here? That was good. That cost me 500 extra euros. This over here is the serpentinization reaction taking place at the off-ridge vents, where the water doesn't come into contact with magma and therefore comes up much cooler. We've got water reacting with inexhaustible reserves of iron 2 minerals in the Earth's crust, and the oxygen remains in the mineral as magnetite and disappears there. The white bubbles come up, they're left over, that's hydrogen. And the yellow sulfur atoms come out as well. Those are sulfur that's always there. This is native metals, and this is what the hydrogen is doing. It's being activated, all right? These are hydrides. This is industrial chemistry. We're not making this up, folks. This is what industrial chemistry does. It works on surfaces, and they're catalytic. These are enzymes, and they give us some very interesting product. They not only give us pyruvate and acetate and formate, they give us some nitrogen-containing compounds that I'm not going to talk about today. And there's a lot of it going on. It's all over the place. And that's why I was talking to Shino about the carbon monoxide on those surfaces. Not all of the surfaces in these catalytic systems are active. There are special sites. That's why it's called heterogeneous catalysis, two-phase system. You don't know what's going on. This gives rise to exergonic reactions flowing downhill energetically. That's what these little arrows are, leading to more stable products, amino acids, bases, cofactors, that not only accumulate in these inorganic compartments, they accumulate at the surfaces by processes known as chemisorption, right? This is just electrostatic interactions between the compounds and the surfaces, and that can allow them to polymerize. And we are very familiar with the ability of minerals to catalyze polymerization reactions, giving rise to more complicated organic compounds, and these have catalytic ability, and that gives rise, in principle, to autocatalytic networks, which we have identified in microbial metabolism. All right, that gives you bases and amino acids, and then it's the standard story, RNA polymerization. Nobody knows exactly how that took place, but it took place. And this gives rise to something like an RNA world, but within the compounds of the little white dots in the little sparks in the background, that's energy release, because there's hydrogen CO2 reaction running in all these compartments, running the system. Okay, put your seatbelt on. It's the origin of translation. We have a ribosome, the genetic code, and protein synthesis. Everybody else has it too. We have it in our compartments that are energetically self-sufficient. Nobody knows the mechanisms, but we know that it occurred in LUCA because it's universal. DNA evolves, we get steps to higher complexity, all being fed by that H2CO2 reaction, just like it occurs in modern acetogens and methanogens, and then off they bubble. The blue acetogens and the red methanogens divide, and it's evolution among free-living prokaryotes, hydrogen CO2 dependent prokaryotes as a starting point for 2 billion years until the origin of eukaryotes later. So that's the movie. We're going to have to go very quickly through. We're just going to have to skip the rest of the talk. What else is there to say? Life is a chemical reaction, an exergonic chemical reaction. It started from a geochemical, an exergonic geochemical reaction, serpentinization. Serpentinization probably takes place on Enceladus. Thank you, NASA, and thank you, Frank Bussberg, for providing me with these slides. This is the E-ring of Enceladus spewing out water because there's water on Enceladus. There's a rocky core, and then probably there's hydrogen coming out from in that water. This is the NASA work, and there are probably hydrothermal vents down there. I don't think there's anything living down there, but if we were to take a methanogen and put it there, it's going to grow and it's going to go quickly. This is just to remind us that this theory ends with real microbial cells, not something abstract, genuine bona fide microbes like they live and grow today. And they're also the kinds of microbes that you can find in these systems. So this is my slide of people who are in the group or have left the group, and they're all smiling because we've managed to get a few good papers throughout the years. These are not all of our papers, but they're some of our papers, and good papers get people nice jobs. Thank you very much for people who did that. This is the group. Many thanks to my friends who are willing to talk to me. And many thanks to my team who has to talk to me because we have to spend the money that we get from taxpayers through these systems. And thank you for your attention. If you have any questions, I'll be glad to take them. If not, that will also be fine. Okay, any other questions?
There's a question here by Eva Stuken. Is it thermodynamically possible for microbes to catalyze the serpentinization reaction?
Almost certainly not, because it's a cubic meter of olivine gives rise to 11 cubic meters of hydrogen, and that is a scale where this process is not going to work. Okay, an acetogen, okay, takes 24 molecules of CO2 and excretes them as acetate for every molecule of CO2 that it incorporates into cell mass. So that means that there's mostly waste product and life is a little bit of a side product. And the serpentinization reaction is too diffuse. And furthermore, when the iron 2 is used up, it's used up. Okay, it has to go back and be recycled through subduction. So the iron 2 is not a catalyst in that sense. It's a substrate. The hydrogen is a substrate, and that's why the serpentinization process is good. It generates massive amounts, continuous amounts of a diffusible substrate that can then undergo catalyzed reactions with other compounds further upstream. It's an interesting idea, but I don't think it would work. Somebody once asked, Dr. Martin, are there organisms that live from pure heat? The answer is yes. They live at hydrothermal vents, and they have chlorophyll, bacteria chlorophyll C, and they live off of 800 nanometer light. Thermal light. They live off of infrared light doing photosynthesis. So it's derived, it's not primitive, but they live off of heat. Yes. Okay. So that's interesting question.
Interesting. Okie dokes.